Hi, in this little video what I want to, hopefully will be short, but you know me, I like to say everything I can help you understand. What I'm trying to do with this little video is to show you and give you the big picture of cellular respiration. Most students tend to get lost on just all the little details of the equation and the reactions and the enzymes, and then they forget how this fits in the big picture, and that's what we need to understand. So you need to go from the big picture to the little thing. You need to know how things work. So remember, as I said many times, that life is just, it's all chemistry. So, but we need to understand what life is all about and what keeps us alive. So I'm gonna do this and also help you hopefully process a little bit more how this relates to everything in your life. So you've seen this chart. This is the basically cellular respiration chart, where you start with the glycolysis right here. We have glycolysis that happens in the cytoplasm of the cell. Cytoplasm, no oxygen required, and it's just the breakdown of glucose in that process. As a consequence of glycolysis, you're going to produce obviously 2 ATPs, as you well know, and that is by the process that we call substrate level phosphorylation. But besides the 2 ATPs that are produced, you also produce 2 NADHs right here, that those 2 NADHs are going to travel, are going to be used at the end, as you know, in our electron transport chain. We also discussed that the final product of glycolysis is 2 molecules of pyruvate, and remember, pyruvate is a 3-carbon molecule, so we have 2 pyruvates. If we have oxygen available, this pyruvate is going to go into the mitochondria, and right in that process is going to lose a molecule of CO2. So this is going to be producing a molecule of acetyl-CoA with only 2 carbons right there. And in the process, if you lose a carbon, you are also going to lose 2 molecules of 2 electrons that are going to be carried by NADH. So glucose gives you 2 NADHs because you release 2 electrons that were picked up by the carrier. When pyruvate comes into the mitochondria, you are going to produce acetyl-CoA, that is a 2-carbon molecule, but in the process, you are releasing electrons. Every time you break the carbons, you release electrons there that are going to be picked up by NADH and carried to the electron transport chain. When this pyruvate that has a 3-carbon goes in, becomes acetyl-CoA. We studied the reaction in class, and you have it in your textbook, and you can follow exactly what carbons are released with which oxygens. The acetyl-CoA is a 2-carbon molecule. Very important that you remember this, that is a 2-carbon molecule now, and it's going to enter the Krebs cycle. And as you recall, in the Krebs cycle, all those carbons are going to be broken off. More CO2 is going to be released. But probably the most important part here in the Krebs cycle is that out of the Krebs cycle, you're going to produce more electron carriers, picking up all the electrons. And you also are, here is where we have the FADH carrier that picks up electrons with a little bit less energy than NADH. It doesn't matter, at the end, electron carriers. Notice that all these electron carriers from glycolysis, from the breakdown of pyruvate, and from the Krebs cycle, all those electron carriers are taken to the electron transport chain. Now, glycolysis, as you recall, produces 2 ATPs. Krebs cycle produces 2 ATPs, all by substrate level phosphorylation. And recall that when you get to the electron transport chain and oxidative phosphorylation, you are going to end up producing about 34 ATPs. A big payoff day. 34, 36, depends on a few parameters, but huge. So total 40. About 40. Now, again, just a nice quick review. Remember, and now I'm going to give you some other important concepts that you should have read, but if not, here we go. Please notice that glycolysis, you are just breaking the energy, the molecule of glucose in the cytoplasm, because you don't use oxygen. No oxygen is needed for this process, and this is the most ancient metabolic pathway in living organisms. Every single organism uses this pathway for the most part, and this is how we are transferring energy from organic molecules to ATP. All cellular respiration always starts with glycolysis, no matter if you are a bacteria or a human, everybody. The only problem with this is that for every molecule of glucose, you are only going to produce 2 ATPs in glycolysis, very inefficient. Now, all prokaryotic organisms, they were the first cells on planet Earth. The first fossil of a prokaryotic cell is about 3.5 billion years old. Prokaryotic organisms, fossils of prokaryotic cells, are about 3.5 billion years old. Right? And an interesting thing that you might not know, but the original Earth's atmosphere had no free oxygen gas. All the oxygen was bound to other molecules forming things like water, carbon dioxide, but we did not have molecular oxygen gas in the atmosphere. So the only metabolic pathway that primitive organisms had or could use to release energy from any organic compound, the only metabolic pathway was glycolysis, because glycolysis does not require oxygen. This is fundamental, very important that you process and digest. Life on Earth first evolved without free oxygen. All the energy captured from organic molecules have to be taken using this process of glycolysis. Alright? And please notice that all cells that do cellular respiration still use this metabolic process. So, guess what happens with the enzymes of glycolysis? We, one of the later organisms to evolve, we share pretty much exactly the same enzymes in glycolysis with the lowly bacteria. The same enzymes are present in the lowly bacteria and us. Exactly the same. We are very related after all. Overview, just to refresh your mind. Remember that we start with we started with glycolysis. We have glucose, 6 carbons. We are going to have a molecule of fructose, 1,6-biphosphate. Don't worry about that. Remember that we need to add ATP to activate, to energize the molecules, to make them a little bit, to prime them, if you wish. After that, remember that the fructose is, when it's unstable, it's going to break into 2 compounds. And the compound that continues the cycle is really G3P, glyceraldehyde 3-phosphate. After many reactions, we end up with pyruvate. These are the same compounds pretty much that you have in the chart that I gave you. Between G3P and pyruvate, a number of things are going to happen that are important. We are going to pick up 2 electrons, and we are going to be able to manufacture a total of 4 ATPs. So considering that you consume 2, you produce 4, the net gain from glycolysis is only 2. But more important, 2 ATPs, that is not much, but the important thing is that we are going to get 2 electron carriers, 2 NADHs, that is very important. And the molecule that it results is 2 molecules of pyruvate. Right? Please recall that every single one of these steps requires a specific enzyme to catalyze this reaction. We didn't mention the enzymes because we don't need to know the names, but you need to know that every single step is catalyzed by a very specific enzyme. So when you get to pyruvate, here is we have our fork on the road. If you have no oxygen, you go into fermentation. If you have oxygen, that pyruvate goes into the mitochondria and proceeds to acetyl-CoA, Krebs cycle, until it's completely destroyed, and that's what we call aerobic respiration. So, pyruvate from the cytoplasm goes in, becomes acetyl-CoA. In the process, you produce an electron carrier. Acetyl-CoA enters the Krebs cycle. When the Krebs cycle is complete, you know well that you're going to produce ATP, you're going to produce CO2, all the CO2 that was contained in the molecule of glucose, all the carbon, but also you're going to produce electron carriers. Now, what's so critical about these electron carriers? What is critical about it? First, all the electrons are harvested and carried to the transport system. All these NADHs and fats are going to be carried to the ETC, the electron transport system, made of mostly those three important proteins that I mentioned in class. NADH dehydrogenase, cytochrome BC complex, and cytochrome C complex. So they are taken to this is the first enzyme, the second, and the third one. These are the three proteins of the electron carrier system. The electrons, when they are released, they are passed from one to another. You know that very well. But these electrons are providing the energy that these molecules, that these proteins need, really, to pump hydrogen ions across the membrane. You are pumping hydrogen ions across the membrane, and at the same time, the electrons are moving from one of these electron transport carriers to another, because at the end of the process, right here, is oxygen pulling these electrons. When the oxygen finally captures that electron that has been moving, the oxygen captures the electron plus the protons that are in the cytoplasm from the regular pH, normal concentration of hydrogen, results in the formation of water. So you actually make water in your mitochondria. That usually is called metabolic water, because you made it from scratch. That's magnificent. You can make water from scratch in your body. Finally, all this work of pumping hydrogen ions, finally you get the payoff when these hydrogen ions diffuse back into the mitochondria through ATP synthase. And as you know very well, ATP synthase, every time the hydrogen ions diffuse through, changes shape and allows for the attachment of another phosphate group into ADP to make ATP. So basically that's the highlights of that, and I hope that this helps you understand what the process is all about. So we needed glucose, we need 6 molecules of oxygen, and you're going to produce 6 molecules of CO2 that are released, and actually 6 molecules of water. That's the metabolic water that we are making inside, and a total of about 40 ATPs. So questions for you, please try to answer them in your head before I give you the answer. Where did the glucose come from? From what you ate, from your intestine to the blood to the cells through the GLUT transport protein with the help of insulin. Where did the oxygen come from? You breathe in, lungs, blood, diffuses from the blood, goes through the cytoplasm all the way to the inside of your mitochondria. Where does the CO2 come from? Breaking the glucose. Every carbon in the glucose molecule is basically released as CO2 from deep inside your mitochondria. So think of the pathway. Mitochondria, cytoplasm of the cell, out into the blood, through the blood, all the way to your lungs. Where does the CO2 go? Up there, to the pool of CO2 in the air. Where does the water come from? The water comes from that oxygen picking up electrons, and electrons negative charge, the hydrogen ions from the cytoplasm join, oxygen plus 2 hydrogens at the end, you have nice water. Those that are the electrons that were carried by all the electron carriers around. The final destination of all those electrons is the oxygen molecule. ATP, you have ATP from substrate level phosphorylation, but most of the ATP comes from ATP synthase, from moving from those hydrogen ions, diffusing from high concentration of hydrogen ions in the intermembrane space back into the matrix through ATP synthase. And that process of ion diffusing is called chemiosmosis. Very good. What else is produced that is not listed in this equation, and very, very, very important? What is produced? Heat, heat, heat. Every energy transformation process, you're going to have some heat lost, energy lost as heat, and cellular respiration is no stranger to that. You are going to have some heat loss, and that's actually the heat that enable us to maintain our high body temperature. So ultimately, the most philosophical question from my point of view, why do you need to breathe? Just to get oxygen, so that oxygen can capture 2 electrons. That's it. That's it. So that oxygen can pull these electrons, but because the electrons are moving, the ATC can pump hydrogen, and because the ATC can pump hydrogen, then you can create a gradient to power ATP synthase. You have to work on this. You have to truly internalize it and make it part of your system. You are living because of this. So I expect you to take the time that you require to fully understand how this works. It's a marvelous machine that works the most beautiful way. So, please remember that the final electron acceptor is oxygen. That's why we breathe. So what happens when you don't breathe? What happens when oxygen is not available? The ETC backs up because nobody's pulling those electrons. They get stuck. It's like in a bad traffic jam. If they cannot move, they get stuck there. There's nothing pulling the electrons, they get stuck. If the electrons get stuck in those ETC proteins, basically any NADH and fats that are made cannot unload their electrons. They stay reduced. And you have a limited supply of these molecules in your cells because you have many other things to take care of inside your cells. So if you cannot unload this, what happens in your cells? All your electron carriers are loaded. And if all of them are loaded, you cannot, all the reactions that keep on breaking down glucose cannot occur because there are no electron carriers available to pick up those electrons. So the process stops. If all the delivery tracks in the world are packed jam and they are never unloaded, can you carry any more things around? No! Same thing here. Oh, that's a good analogy. So obviously ATP production stops because ATP synthase doesn't get the concentration gradient needed to operate. No ATP, no energy, and that's the end. So please consider really why you breathe and why this is so important. This is so fundamental that you understand about your life. So that brings me to the case of what happens when you don't have oxygen, when you stop breathing, or when you take compounds like cyanide that block this process. When that process is blocked, you die. That's what cyanide does. Alright? So after that deeply philosophical concept that what happens when you don't have oxygen. Not all is lost. There is hope. There is hope always. There is hope. Always. There is hope. Remember that. So, you have pyruvate. If you have oxygen, oh my God, we go into the mitochondria and we produce at the end a total of 40 ATPs. Life is wonderful. What happens if you have no oxygen? Sometimes we have that problem. No oxygen. That is going to be fermentation, and I'm going to try to give you a highlight, even though you are totally able to figure out on your own, but just to reinforce your concepts. So, main problem with no oxygen. Your cell is loaded, all the electron carriers are loaded, and they cannot be reused anymore. So you are stuck. So here we have the second step after you make pyruvate and all the carriers are loaded. We have hope. And that hope is called fermentation. And we have alcoholic and lactic acid. So let's start with what we call alcoholic fermentation. Alcoholic fermentation. Who does this process? This process is done by yeast, a type of fungi, unicellular fungi, and a few bacteria. But what do you have to remember? Yeast. Alcoholic fermentation, yeast. What do yeast do with that pyruvate? If there is no oxygen around, yeast grab the pyruvate and through a couple of chemical reactions that I'll show you, remove one carbon and produce a wonderful molecule called ethanol, plus the carbon that they removed. What does this process accomplish? You will see this process can only happen because in this step here, we are going to use one of those loaded NADHs and we are going to unload it to make ethanol. And this is wonderful because now this NAD plus, the oxidized form, can go back and pick up a few more electrons when you break glucose. So if nothing else, you can keep on breaking down glucose and releasing 2 miserable ATPs. But 2 is better than nothing. So this process, all this process does, from pyruvate, you lose some electrons when you break this carbon off, and that allows to, or consumes 2 electrons, and that allows us to recycle NAD plus so it can go back to glycolysis to continue the breaking down of glucose. If nothing else, 2 ATPs is better than nothing. And that's what yeast does. And that's the process that we use basically to get beer, wine, and bread. Bread. When you make bread, what do we know happens in bread? Gets puffed up. What makes it puffed up? The CO2 that you produce. That's what all those little bubbles inside your bread is. The yeast in the dough is using the glucose, is doing glycolysis actually with all this starch in the flour, and is producing bubbles of CO2 that make your bread puff up. It's also producing ethanol. So why don't you get drunk when you eat bread or any other baked good that has yeast? Because when you bake it, the ethanol evaporates. And that's that beautiful smell that you smell in your house when somebody is baking bread. Oh, that's wonderful. Thank you, yeast. Now, you, animal, me, animal, and some bacteria, some awesome bacteria, do a different type of fermentation called lactic acid fermentation. Because, again, we start with pyruvate. Notice, no oxygen. Hello. We start with our pyruvate. And we can transform that pyruvate if we add an electron. Unloading, we can transform that pyruvate into lactic acid. And that allows us to recycle our NAD+. Now this lactic acid, again, most wonderful thing, because allows your muscles to keep on going for a little bit, sprinting full time. You cannot sprint forever, because only 2 miserable ATPs are not going to power you forever, but good enough to get to the end of the 100 meters. But the most beautiful thing is that this lactic acid is basically what you have, provides the acidity to make kimchi, sour cream, yogurt. That's why they are sour, because you have acid there from lactic acid. So the bacteria, for example, use all the sugar in milk, the sugar is galactose, and transform that sugar into pyruvate, and then since they don't have oxygen, pyruvate to lactic acid, and that's how you make yogurt. Same thing in your muscles. You break down glucose, you produce 2 miserable ATPs that allow you to keep on running. When you get to pyruvate, in order to recycle your electron carrier, your pyruvate picks up those electrons and becomes lactic acid. But you start accumulating lactic acid, pH of your cells changes a little bit, and only 2 ATPs at a time, it's not going to power you through very far. So that is anaerobic exercise, the ones that you do when you sprint or when you lift weights. That's you maximize the power but very short time. All anaerobic exercise. Alright, so I want to show you the details of this. You can look at it in the textbook, but again, sometimes it's hard for you to see these things. We said that pyruvate to ethanol and CO2. So looky, please focus this and pay attention. So you have your glucose, and we know in glycolysis you're going to produce 2 ATPs, but you also produce 2 NADHs here. And the product is pyruvate. We're going to add the hydrogen there to pyruvate. What's going to happen to pyruvate? All this catalyzed by enzymes. Notice pyruvate is going to lose a molecule of CO2. It's going to lose a molecule of CO2. So it's going to lose a molecule of CO2. And then after it loses the molecule of CO2, you have this molecule. CH3, same thing. CO right there. And the hydrogen that is there. What you lost was this CO2 right here. This molecule is called acetaldehyde. Don't worry about the name, because this is a short-lived molecule, because pretty soon, guess what's going to happen to this acetaldehyde? Check it out. CH3COH is going to pick up those electrons and unload your carrier. Notice where the electrons go. Please, notice where the electrons go. This is the original molecule. We have CH3 here. We have our carbon there. We have an OH there. OH. You just gain hydrogen. You just gain electrons. These electrons were given to you by the NADH. This is ethanol. This is ethanol. So the only thing that you're doing is unloading this in the form of hydrogens, and the final molecule that is produced there is ethanol. 2 carbons in ethanol, because the other carbon was lost as CO2. Please follow this in your textbook and just look at the molecules. It makes so much easier to understand when you know where these oxygens and carbons are going. Lactic acid fermentation, exactly the same process. Please, please, please. Goal is to recycle the NADH. Now, look at this, please. From glucose to pyruvate. Again, I like to show you the non-ionized form so you can see basically all the atoms. That's an OH there. This is your pyruvate. Notice what happens to this pyruvate. Notice what happens to the pyruvate. I can do it with the H there. Notice what happens to the pyruvate. What's the difference between pyruvate and lactic acid? Again, what's the difference between pyruvate and lactic acid? Compare. CH3 right there, same thing. CO, OH. What's new in this molecule? What is new is that hydrogen there. That's what makes it new. And where this extra hydrogen come from? From NADH unloading it. That's the goal of fermentation, basically to unload NADH So now this NAD plus can go back and allow you to keep on breaking glucose for as long as you can. Even though it produces 2 ATPs, it's better than nothing. It allows you to continue living and performing your functions for a little time. Please notice, all this lactate or lactic acid is moved to the liver, and your liver breaks it down and recycles it again. It's very amazing. So here you have another job that your liver does. Your liver detoxifies your drugs. Your liver stores glycogen. Your liver is going to break down the lactic acid that you produce during intense anaerobic exercise. So, aerobic exercise, I can breathe and I can do this level of activity for hours. Think of a marathon runner, between 2 and 6 hours running and no problem, because your mitochondria are getting enough oxygen to do this. If I try to sprint, however, the amount of energy required, there is no way I can deliver enough oxygen to my mitochondria, and my cells are going to have to use some anaerobic respiration for that. That's it. Now, important thing for all the athletes. When you train, when you're exercising, part of the training process, what it does is increases the number of mitochondria in your cells. Early in the season, when you have to run, your breathing rate is extremely high because you need to maximize the amount of oxygen that you get. As you practice more, your cells get the message, man, we don't have enough mitochondria. What do we need to do? We don't have enough mitochondria. Mitochondria divide and produce more mitochondria inside your cells. So what happens towards the end of the season as the number of mitochondria inside your cells increase? Basically, you are more efficient at using oxygen because you have more mitochondria, you can produce more ATP, they can remove the oxygen from your blood more efficiently, higher rate of diffusion. So towards the end of the season, you can run the same speed, but instead of breathing so hard, you can breathe much more slowly because the process is much more efficient because you have more mitochondria. That's what training does to you, basically. Increases the number of mitochondria in your cells. The season ends, you go home, you sit in the couch for a month. Guess what those cells do with the extra mitochondria? We don't need all this mitochondria. Let's use these compounds for something that we need. And you get rid of your mitochondria if you don't exercise. That's why you lose all your fitness level at the end of the season if you don't continue working on it. Make sense? The other question that somebody asked me, and I want to address it, I said the lactic acid goes back to the liver, is transported to the liver, to the blood, by the blood, to be broken down and processed. That's why after a heavy exercise, one of the things that they ask you to do is to cool down. Either jog slowly around for half an hour or so, or keep on active. Don't just stop cold. Because when you are moving, your blood is circulating much faster and is helping carry all this lactic acid that accumulated in your cells back to the liver. Basically, it's cleaning or clearing your cells of lactic acid. That's why if you move, it's less painful the next day. So you are still going to be in pain because you break the fibers in your muscles with intense exercise. But the idea of this cool down process after exercising is to help transport, speed up the flow of blood, and remove as much CO2 from your, not CO2, lactic acid from your cells to take it to the liver. Just practical application. So if you're out there exercising, please think about these things when you are doing it. It helps stick it into your brain. So the last thing I want to bring to you, again, applications, cellular respiration and what controls it. You can have a number of enzymes that are involved in the process of cellular respiration. The way cellular respiration works, if you produce too much ATP at the end, the ATP actually blocks, the excess ATP blocks one of the early enzymes in the process and stops this. Remember, you cannot accumulate ATP, so there's no point on doing something for nothing. Your cells are not wasteful. So, ATP actually, one of the products of cellular respiration, is an allosteric inhibitor. Those are the ones that attach to the allosteric site of an earlier enzyme and stops cellular respiration. So, you don't produce more than what you need. It's an on-demand service. So let's look at this. This is feedback inhibition. You're familiar with this now. On the very top there, I have glucose. I'll try to highlight it. I have glucose there. Then you know glucose, you are going to produce at some point through a number of reactions, you are going to get down to pyruvate. Yes? I'm going to switch pens. One of the intermediary products is one called fructose 1,6-biphosphate right here. This is during glycolysis. And the enzyme, this is the one that you need to know. The enzyme between, one of the enzymes is called phosphofructokinase. This is one of the enzymes in glycolysis. Enzyme. And it's called phosphofructokinase. You can look it up in your textbook, in that where we follow glycolysis, so you see where it's at. You don't need to do anything else, just look at it. What happens to phosphofructokinase? Pyruvate, pyruvate to acetyl-CoA, Krebs cycle, electron transport. You produce a ton of ATPs there. If you are producing too many ATPs that they start accumulating in your cell instead of being used to move or think, this excess ATP attaches, the ATP molecule really attaches allosterically to phosphofructokinase and changes its shape. And when phosphofructokinase changes shape, it cannot work and blocks the whole process. So ATP inhibits phosphofructokinase allosterically, attaches to allosteric site. Notice what else blocks phosphofructokinase? Citrate. That's the first compound produced in the citric cycle. When acetyl-CoA joins that 4-carbon molecule, if you have an excess of citrate or citric acid being produced, because this is feeding too much acetyl-CoA here, it also is going to inhibit the enzyme allosterically. So it's a beautiful system that allows you for the regulation of ATP production. The products inhibit or stop the process. Now, what happens if your amount of ATP decreases because you're exercising or doing something? Basically, there is no ATP, gets removed, and phosphofructokinase gets to go back to normal and continue the process. Beautiful, isn't it? So please, this is how it works. Usually excess product stops a metabolic pathway, and that's how you regulate it. You're gonna see many, many examples of this through the year, but please get it down now. Basically, your cells do not do work for nothing. They can regulate just like a productive factory. If you are not selling your products, the factory closes or stops or slows down the process. Same process inside your cells. And again, ATP inhibits phosphofructokinase. Citrate also inhibits the enzyme to stop the cycle. Now, we talked about the process of cellular respiration. Glucose is the favorite substrate because it's the one that goes the easiest in. However, there are other things that can be used to power your cellular respiration. And you know that mainly if you go into some of these low-carb diets. Of course, you're still alive and you're still doing cellular respiration. So I want to show you this so you get a picture, and every time you eat, you can think what's happening to all the stuff that we eat. Any polysaccharide that you eat, starch or glycogen that's stored in your in your liver, any sugars basically, through some changes, they're ultimately going to become basically glucose. Hydrolysis of starch produces glucose, hydrolysis of glycogen produces glucose. If you have any other sugars that you have consumed, galactose from milk, fructose from fruits, any other type of sugar, basically somehow they're going to be ultimately always modified into glucose to feed glucose to the glycolysis. So they are all modified inside your cells. You have the enzymes inside your cells to do this. What happens with your proteins? If you don't have enough carbohydrates to power this, guess this is what happens to proteins. You know, proteins are broken down into amino acids. You know the structure of your amino acids there. What's going to happen? The central carbon with the R group, oh, sorry, my lines are a little bit off, are going to be used. The amino part, this amino part, the part with the nitrogen, is not good for you. This part, the central carbon and the R group, is going to go into glycolysis or the Krebs cycle, and we'll see where it enters. Depending on the amino acids, you can have amino acids with 2, 3, 4, 5 carbons. Depending on the number of carbons that they have is where they're going to enter the whole process of cellular respiration. Keep that in mind. Number of carbons, important. This nitrogen, that is basically eliminated as waste. This is what's going to become ammonia, or urea in most of our cases. And this is what we eliminate with our urine. We pee the ammonia. Most of the ammonia comes from eliminating the amino group from the amino acids. And actually, I want to add something important again. This is released by your cells, the NH2 group, as ammonia. Flows through your blood. Ammonia is toxic. It's going to kill you if you don't have enough water. So guess what your liver does with ammonia so you don't die? Your liver transforms your ammonia. Your liver transforms this molecule. Your liver again. Hello, liver. Thank you, liver. Your liver is going to transform this molecule into a compound called urea that is not toxic, so you can pee it safely. Urinate. In your urine, this is what you are releasing for the most part, and that comes from this. But that's another story. The focus here again, sorry I got sidetracked, I just love the liver, is that depending on the number of carbons that your amino acid has is where it's going to enter the different parts of cellular respiration. Now this is the scary part. You know fats, right? Mostly the triglycerides. Mostly the triglycerides that are made out of glycerol and fatty acids. Glycerol is a 3-carbon molecule. Guess what? Enters glycolysis where G3P is. Glyceraldehyde 3-phosphate, glycerol, they are very similar molecularly. So this enters right there, straight to the glycolysis. Guess what happens to these long chains of fatty acids? Ready? This is scary. They are broken up. Broken up. Let's make it more dramatic. Ready? Right there. Check it out. What's happening? Each one of them is breaking out into twos. 2-carbon compounds. And if you recall from the cellular respiration, the molecule with 2 carbon compounds is acetyl-CoA. So this, a fatty acid breaks, your cells break them into 2 carbon compounds, and these 2 carbon compounds are fed into the Krebs cycle as acetyl-CoA. And all these hydrogens, hello people, all these electrons that you see here are gonna go straight to the electron transport chain carried by NADH when it's reduced, and it's gonna produce a truckload of ATPs. Lots of energy from these molecules. It's beautiful. So, summarize, you have this in the back of your big, humongous sheet that I gave you. So let's look at it. Check it out. More than the words, I don't care so much about the words, but I care about that you understand these diagrams. So let's just do it the old-fashioned way. Again, if you have carbohydrates, any type of carbohydrate, the sugars are basically going to go into glycolysis. The favorite sugar, of course, is glucose. That goes into glyceraldehyde, pyruvate, acetyl-CoA, Krebs cycle, electron transport chain, and track load. So carbohydrates, you know the process. Now, if it's a sugar with a different number of carbons, you know, some sugars have 5 carbons or some sugars have 3 carbons, well, they're going to enter at these different places here in the glycolysis process. Now, what happens with proteins? Remember what I told you about proteins? Proteins are broken down into amino acids by your digestive system. When the amino acids get to your cell, the amino group is released, and this amino group, your liver actually is gonna combine, NH3 is 3, your liver is gonna combine several of these to make a compound called urea, and that you basically release in your urine. That's the amino group. The rest of the amino acid, depending on the number of carbons that your amino acid has, is gonna enter at different places. If it has 3 carbons, replaces pyruvate, technically. If the amino acid has 2 carbons, replaces acetyl-CoA. If your amino acid has 4, 5, or 6 carbons, it's gonna replace one of the molecules here in the Krebs cycle. So you can use anything, anywhere. Your cells are just wonderful machines, multitasking. And then from there, of course, you know that you produce a lot of electron carriers that are fed here. That's what you do with proteins. Basically, you can use them also there. What's the last group that we need to process here? The last group is your fats. And you know fats are made out of glycerol and fatty acids. We said glycerol because it has 3 carbons, basically enter there in the place of glyceraldehyde 3-phosphate, G3P. Remember that this is G3P. What happens to the fatty acids? All the fatty acids are broken into 2 carbon pieces, and each of those carbons is loaded with hydrogen. These 2 carbon pieces go in the place of acetyl-CoA, because remember that acetyl-CoA is also a 2 carbon molecule. Acetyl-CoA has 2 carbons. So these go in the place of acetyl-CoA, and since each fatty acid has more than 100 of these, it's just an unbelievable number of acetyl-CoAs that power the Krebs cycle to produce a truckload of electron carriers to produce a truckload of ATP. So as you see, you can go anywhere, and that's why you can use, remember what we mentioned at some point here, that the first thing that you use for energy are carbohydrates. When your body runs out of carbohydrates, including glycogen, if you don't have any more glycogen stored after carbohydrates, you use your fats. And again, part of the training during exercise is to increase, the training requires your body to increase the number of molecules, enzymes basically, that you use to process fats. So when you are training in any sport activity, you are not only increasing your mitochondria, but you are priming your cells to increase also the number of enzymes needed to break down fats. So when you really need to use that, by the end of the season, you have all those molecules in high quantities, so you are very efficient at it. So, carbohydrates, fat. What's the last thing that your body uses as a last resort? Your body uses as a last resort proteins, because proteins really are, the purpose of them is to use them to build the proteins that you need for your body to work, from muscles to enzymes to tendons, ligaments, all that stuff. So your body proteins come in the third place. All right? So I hope that this helps you understand how your body works, how what we eat, anything that we eat basically feeds into the electron transport chain one way to another, and is all used. Now, some of us tend to sometimes eat more than what we need to, and again, our body is so clever that when you eat more than you need to, doesn't matter if it's carbohydrates, doesn't matter if it's proteins. If I overload my body with proteins or carbohydrates or anything, ultimately your body is not going to get rid of them because it's a valuable molecule. So if you overload your body with those molecules, your body, instead of using them here, if you don't need them, all these processes that feed here can be reversed actually, and all the processes go back to fat. So these are the series of reactions that keep you going, that make your body do whatever they need. Alright, so I think I have reached the end. I hope this helps you understand a little bit more how your body works. Make you more aware of what you eat and what happens to everything that you eat. And again, as I said before, if you think about these things every day when you're doing them, they stick to your brain and they'll be with you for life. And share them. Teach your parents about this. Just show them. A lot of them, if they are my age, we don't know a lot about this because we didn't learn this in our old schooling. It's new stuff that we have learned since then. So please share it. Once you teach somebody about this, it really gets into your head. Thank you, guys.